Okay, so welcome back to this next video in which we're discussing the glycine receptors and their involvement in hyperekplaxia. Right, okay, so we were just discussing which way chloride anions are going to move through this open glycine receptor now, whether they're going to move in or out. And we've discussed basically that the concentration gradient is favouring the movement of chloride anions in. It's about a 30-fold gradient favouring the movement of chloride anions in. However, what is the electrical potential difference across the cell membrane? So we discussed that the electrical potential difference across the membrane really means the change in the electrical potential that your little machine will measure if you move from extracellular to intracellular. And this is usually, in most neurons, around negative 65 millivolts. So what does that mean? That means that the electrical potential intracellularly is around 65 millivolts lower than the electrical potential extracellularly. Okay? Now, chloride anions are negatively charged chemical species. They want to be where the electrical potential is higher, which in this case is the extracellular compartment. So they want to be in the extracellular compartment. Okay? So basically the electrical gradient is favouring the movement of chloride anions out of the cell. Now the question then is, which one wins? Does the concentration gradient, which is trying to move chloride anions into the cell, or the electrical gradient, which is trying to move chloride anions out of the cell, win? And the answer is that the concentration gradient wins. So the chloride movement out of the cell, because of the electrical gradient, is smaller than the chloride movement into the cell. So overall, the concentration gradient is stronger than the electrical gradient. So you get a net movement of chloride into the cell. It's vastly reduced from what it would have been if you just considered the concentration gradient, and if the two electrical potentials, i.e. the electrical potential extracellularly and the electrical potential intracellularly, have been the same value, i.e. there was no electrical potential difference across the membrane, then it's nowhere near as big as that movement would be, but there's a little movement now of chloride anions into the cell. Okay, now when you bring chloride anions into the cytoplasm of the cell, what's going to happen? Well, chloride anions have a negative charge, so you are bringing in negatively charged ions into the cytoplasm of the cell. In addition, you are removing them from the extracellular space. You're not just coming up with them from nowhere, you are bringing them in from the extracellular space. So, you are bringing in negative charge from the extracellular to the intracellular. Now, bringing it into the intracellular the intracellular compartment will reduce the electrical potential intracellularly. That's the effect of putting more negative charge here. It'll lower the electrical potential. In addition, you are removing negative charge from the extracellular compartment to bring it into the intracellular compartment. So when you remove negative charge from the extracellular compartment, that's going to have the effect of raising the electrical potential there. Okay, because negative charge brings down the electrical potential. Okay, so when you remove it, that's going to lead to the electrical potential going up. So the electrical potential extracellularly is going up, and the electrical potential intracellularly is going down. The way we started was that the intracellular electrical potential was 65 millivolts lower than the extracellular electrical potential. If this one's going up and this one's going down, then the amount by which the intracellular electrical potential is lower than the extracellular electrical potential is just going to get greater, i.e. the voltage from extracellular to intracellular is going to become more negative. Now when the electrical potential difference across the cell membrane becomes more negative, we call this hyperpolarization of the cell membrane, because the polarity in electrical potential across the membrane is becoming greater. Okay, so hyperpolarization, and we say that the membrane is hyperpolarized. Okay, so the effect of bringing negative charge in is to hyperpolarize the cell membrane, and therefore the effect of opening these glycine receptors is to hyperpolarize the cell membrane. Now, what does this mean for our motor neuron in our spinal cord here? Well, basically, in order for the alpha motor neuron to fire, it needs to depolarize the electrical potential. The electrical potential needs to become less negative, i.e. the amount by which the intracellular electrical potential is lower than the extracellular electrical potential needs to become less. This is having the opposite effect, basically. So it's going to make this alpha motor neuron less likely to fire. And basically there will be other neurons synapsing onto this alpha motor neuron. And I don't mean to imply that it's coming out of there, but there will be another neuron, let's say here, which will have an excitatory synapse with this alpha motor neuron. Okay? And excitatory synapses will be allowing positive charge to move into the cell. So let me just give you a bit more terminology here. So, when we open these glycine receptors here, we are getting this movement of negative charge across the cell membrane in the form of chloride anions. Now, when you move charge, that's known as a current. So this movement of negative charge across the membrane through the glycine receptors is a current. Now, it's inhibiting the alpha motor neuron. It's making it less likely that it's going to fire an action potential. So this current that comes through the glycine receptors is what's known as an inhibitory postsynaptic current. Inhibitory postsynaptic current. And basically, inhibitory postsynaptic currents are often abbreviated to IPSCs for short. This is an IPSC. Right, so glycine activating these these glycine receptors is going to trigger inhibitory postsynaptic currents of negative charge being brought into the cell. Now, these excitatory synapses here, so I drew another neuron here, which has an excitatory synapse. Well, I just declared that it was an excitatory synapse with the alpha motor neuron. This is going to be releasing some neurotransmitter, maybe glutamate, onto the alpha motor neuron, and it will result in a current into the cell. So you'll open a channel and it will allow a current into the cell. However, this current will be positive charge coming into the cell, usually sodium ions. Okay, and when you've got positive charge moving into the cell, that will depolarize the electrical potential of the cell membrane, because you will raise the electrical potential intracellularly and lower the electrical potential extracellularly. So the amount by which the intracellular electrical potential is lower than the extracellular electrical potential will go down, so the voltage across the membrane will become less negative. So it will make it more likely to fire an action potential, and it's therefore referred to as an excitatory postsynaptic current, or an EPSP. So basically, this alpha motor neuron will have both inhibitory postsynaptic currents and excitatory postsynaptic currents coming into it across its membrane. Okay, now the excitatory postsynaptic currents will be making the alpha motor neuron more likely to fire, and the inhibitory postsynaptic currents will be making the neuron less likely to fire. So basically, they might just end up neutralizing each other, i.e. the effect of moving this negative charge in might neutralize the movement of the positive charge in, and the alpha motor neuron might overall do nothing. Okay, so the point of all of this is that if for some reason the glycinergic neurotransmission went down, so that's an interesting adjective that you should know the name, so glycinergic. When we're talking about neurotransmission involving glycine, people often use this word glycinergic to describe the neurotransmission. So, if for some reason we were to reduce the glycinergic neurotransmission onto alpha motor neurons, i.e. reduce the amount of glycine or the effect of the glycine on the alpha motor neurons, then what would happen? Well, the IPSCs would go down, so you'd get less negative charge coming into the cell, and this would then mean that the excitatory synapses from these other neurons, those would no longer be neutralized, basically. Their EPSCs would no longer be neutralized by the IPSCs of the inhibitory neurons, okay? So that would mean that these EPSCs would now potentially be able to stimulate the alpha motor neuron to fire an action potential, and that would lead to the overfiring of the motor neurons. And if they overfire, what's going to happen? Your muscles are going to contract too much, so you might get convulsions, you might get a constant contraction, basically. You might get muscle stiffness, which is when the muscles are just continuously contracting. Okay, so drops in glycinergic transmission can be problematic, and we're going to see that this is what happens in hyperekplaxia. Right, okay, so what we'll do now is we're going to look at the structure of the glycine receptor in a bit more detail. Okay, so the glycine receptor is a member of the family of ligand-gated ion channels known as cis-loop ligand-gated ion channels. Okay, so there are other very famous members of this family, members that are far more famous than the glycine receptors. So other famous members of the cis-loop ligand-gated ion channels are the nicotinic acetylcholine receptors, the GABA A receptors. They're the two biggies, they're very, very important. Then slightly less famous is the 5-HT3 receptors. Okay, and then this final family is then the glycine receptors. And I want to stress that these are whole families of receptors. Each one of these four members here, they're not a single receptor, they are families of receptors. Okay, so we are going to study these glycine receptors. So let's see what the structure of a cis-loop ligand-gated ion channel. And this actual discussion that I'm going to say now, there's nothing here that's specific for glycine initially. It will be obvious when I suddenly start talking about glycine, but what I'm initially going to talk about is true for all of these cis-loop ligand-gated ion channels. So, let's say this is the phospholipid bilayer here, and then we've got our cis-loop ligand-gated ion channel here, which we'll say is a glycine receptor. Okay, so here's our glycine receptor here, and it's in the phospholipid bilayer. Now, it is after all an ion channel, so it needs to have a pore down the middle through which the ions can move. Now, the question is it made up of a single protein, or is it a whole complex of proteins stuck together? And the answer is it's 5 proteins stuck together. So a glycine receptor, often abbreviated to GlyR, is not just one protein. It's 5 proteins stuck together. Okay, so here's 1, 2, 3, 4, 5. So it's what you would call a pentamer. It's made up of 5 constituent subunits. So it is a pentamer. Right, okay, so let's now take out one of these subunits that makes up the pentamer. So let's take this one here out and have a look at its membrane spanning topology. So let's have a look at this polypeptide in a bit more detail. So here is the phospholipid bilayer drawn again, and now let's have a look at this protein in more detail. Okay, so we start with the amino terminus of the protein, which is on the extracellular side of the membrane. So this is the extracellular fluid, the ECF here, and then this is the cytoplasmic side here. So this is the cytoplasm. Okay, right. So the amino terminus, on the extracellular domain, you then also have a very important thing as far as the naming of this is concerned, which is this cis loop. So you have this loop in the polypeptide structure that is held together by a disulfide bond that is formed between 2 cysteine residues on the opposing strands. And don't worry, I'll talk you through what a cis loop is in a bit more detail in a moment. Then you have 4 membrane-spanning alpha helices. So here's the first membrane-spanning alpha helice. Here's the second, the third. Then you have a large loop between the third and the fourth membrane-spanning alpha helices. And then here's the C-terminus of the protein. So basically, it straddles the membrane these 4 times. So let me highlight some important portions of this structure. Okay, so we'll start with the 4 membrane-spanning alpha helices. So in turquoise, these are the 4 membrane-spanning alpha helices, and they are labelled M1 through M4. Okay, so this first membrane-spanning alpha helix is M1, then you're followed by M2 here, then M3, and then you have a large loop before you then get to M4 here. And this large loop is the next portion I want to talk about. Okay, so the M1 to M4 membrane spanning alpha helices, they make up the transmembrane region of our glycine receptor subunit. Okay, then we have this large intracellular loop here, also known as the M3-M4 loop, but more commonly just called the intracellular loop. Okay, so this is the intracellular loop, or you can also call it the M3-M4 loop, right? And this basically makes the intracellular domain of the glycine receptor. Now finally, you then have this extracellular domain here, which I'll colour in orange. So this is the extracellular domain of our glycine receptor, and this makes up the extracellular domain, okay? So dominating this structure is this cis loop, which is this loop in the polypeptide structure held together by a disulfide bond between the phyl groups of cysteine residues on opposing strands, and I will discuss the structure of a cis loop in more detail in the next video.